Hello, my name is Amy Dykstra, and I'm going to talk to you about my research involving the Echinacea project. And so here we go. So my research looks at various aspects of habitat fragmentation and how habitat fragmentation affects native prairie plants, specifically Echinacea angustifolia. So just a definition of habitat fragmentation. Habitat fragmentation is the loss of habitat as habitat is converted from native habitat to something else like cornfields or human habitation. And so these are some classic diagrams here showing how woodland in Wisconsin was fragmented. And so you can see over here on the left in 1831, the area of a certain township in Wisconsin was almost completely woodland. And as that forest was converted into agricultural fields, the native woodland was chopped up, and a lot of it was converted to some other use. And by 1950, the remaining habitat is reduced to these tiny little green spots that are separated from each other, isolated from each other, and are also very small. And this has happened with prairies in the Midwest. And here's a map of Minnesota that shows the historical prairie as kind of the yellows and tans in this figure. And then what's left of the native prairie now is shown in these red dots. And so much of the native prairie is gone now. And what's left is just small fragments of native prairie. So one consequence of fragmentation of habitat is that native populations of plants and animals are reduced to very small remnant populations. And this can have negative consequences for these populations. One negative consequence is genetic. So if you have very small populations, it's likely that individuals are mating with close relatives, and this can lead to inbreeding depression. Also, small populations are sometimes subject to more genetic drift. So chance changes in their in the genetics of the population, in the gene pool, just based on chance. So certain alleles can be just randomly lost from the population based on chance. There are also demographic effects, what I call demographic effects, and this is an increased vulnerability of small populations to just chance events. So maybe a flood will wipe out an entire population if it's a small population, where a larger population, there would be some parts of its habitat that would not get flooded, for an example. So my research questions that have to do with the effects of habitat fragmentation on plants are, number one, whether small remnant populations are actually declining as we might expect. Number two, if these populations are genetically depleted because of genetic drift or inbreeding depression, can we rescue these genetic declines by outcrossing to other populations? And then thirdly, are these fragmented native populations locally adapted to their particular area compared to another area farther away? So the study species that I used is Echinacea angustifolia, which is known as commonly as narrow-leaved purple cone flower. We think that Echinacea represents other species that are native to the tallgrass prairies. In some of its characteristics, it's a long-lived perennial. It reproduces by seed and not vegetatively. It's pollinated by generalist insects. It doesn't have a specific mutualism with a specific pollinator. It is self-incompatible, so that affects its ability to reproduce. If it is close to very close relatives, it may not be able to reproduce. And it rarely flowers before the 7th year. Every year it grows back from a taproot, and it may not flower for several years. It may just be a vegetative plant for several years. The study area is in kind of west central Minnesota in the southwest corner of Douglas County. And if we expand a little bit on this map, this is showing our study area, the southwest corner of Douglas County again. Over here, these purple hatched areas, this is the town of Hoffman, this is the town of Kensington, and all of these red squares that you see on the map are remnant prairies that our group studies. And I've had the happy ability, the fortunate ability to participate with the Echinacea project. So my first question was whether native Echinacea populations within this study region are actually in decline. So as you can see in this picture, there are several Echinacea plants, and they tend to be relatively common in tallgrass prairie. And but we know that their populations are fragmented. So I wondered if these populations are actually growing or staying stable or declining. And then secondarily, I wondered what role the seedlings play in population dynamics. So most of the aspects of my research, at least initially, kind of focused on the seedling stage of the life cycle. And I wanted to know whether small remnant populations are declining more rapidly than larger populations. So this research built on an annual search for seedlings that our group did from 2006 to 2013. We studied 14 prairie remnants, and in each of those remnants, we would identify plants that we knew flowered the year before, and we would search within about a meter square around those plants. And then also we searched some random locations kind of to get an idea of how far seeds are dispersed. And so we were able to find seedlings that we were able to recognize because they still have their persistent cotyledons, and showing that they just germinated, and then they have a very distinctive first leaf that is quite fuzzy. So it's very distinctive from many other kinds of seedlings. And we carefully mapped these seedlings, and over the course of the 6 or 7 years when we searched for seedlings, we found a total of 914 seedlings. And as of 2019, 87 of those seedlings were still alive from all of the different years. There have been a few of these plants that have survived long enough to flower. We've had a lot of mortality, but one plant in particular at Stevens Approach flowered in 2013 and in 2014. And it was mowed both of those years, so it did not contribute any seeds to the next generation. And then there was also a plant at EELR, East Elk Lake Road, that flowered in 2019, and it may have flowered another time. And so that one possibly contributed seeds. So By site, you can see the site designations over here, and you can see that there's a lot of variation in where we found seedlings from year to year. There's also variation from year to year. There's some sites, this Stevens Approach site, where we found seedlings every year. There are other sites where we did not. Well, I'll point out landfill. We found a large number of seedlings in 2008, but no seedlings in 2006, 10, 11, 12, 13. So a lot of year-to-year variation and site-to-site variation. This is a table of the seedlings that were still living in 2017. I probably should have updated this for 2019, but I didn't. And you can see that we still had one plant that was still surviving that we had found initially in 2006, and several that we initially found in 2007 as seedlings, a whole bunch that we found in 2011, and we are still tracking these seedlings. So in addition to these seedlings that we've been tracking all this time, in 2009, I also went out and found a bunch of basal Echinacea plants and a bunch of flowering Echinacea plants. In fact, I found a total of about 800 basal plants and about 69 flowering plants. And then I returned in 2010 to see whether these plants had survived, whether the basal plants had started to flower, whether the flowering plants were flowering again. And so this is the distribution of data. This is the status in these columns that they were in 2009. There were 44 of the seedlings that survived and were basal in 2010, and 21 of them we couldn't find, and we presumed that they had died. And then a distribution also of the basal plants from 2009, 146 of those we couldn't find. 3 of the flowering plants we couldn't find again in 2010, but most of them we found, and about half of those flowering plants were flowering again in 2010. So this is a simplified life cycle graph. The S here would be the seedling stage, and these seedlings would, if they survive, they become small basal plants, and then usually they grow bigger until they reach a size that's large enough to where they can flower. So we've got small basals, larger basals, even larger and even larger basal plants, and then finally flowering plants. And so many of these stages plants can be in for several years at a time. So a small basal plant might stay as a small basal plant for a year or 2 or 3 or many years. And sometimes a larger basal plant, when it comes back the following year, is actually smaller. It has fewer leaves than it had the year before. And sometimes they can kind of skip over from stage to stage. So as I showed you on the previous table, about 2/3 of the seedlings survived from 2009 to 2010 and became small basal plants. And of those small basal plants, about 2/3 of them survived and stayed as small basal plants. A few of them grew larger. One of them grew quite a bit larger, and apparently at least one of them flowered. And so if we filled out all of these transitions, this life cycle graph would start to get pretty complicated. And so it's a little bit easier to show this in a matrix. And so again, in this matrix, the columns would be the status in 2009, and the rows would be the status in 2010. So again, here are those seedlings that were seedlings in 2009, and then they were basal plants in 2010. And these larger basals especially are more likely to become flowering plants in the following year. The flowering plants, about half of them were flowering again. Some of them, when they came back as basal plants of various different sizes. And then this cell here shows the number of seedlings that were produced by each flowering plant. So each flowering plant that we searched in 2010, about 1 out of every 4 of those flowering plants that we searched had a seedling. And so each flowering plant produced about a quarter seedling. So the purpose of setting up this matrix, which is called a population matrix, is to get an idea of what the overall population growth rate is. And you can do some simple matrix algebra, or at least it's simple if you put it into a program like R. And if you calculate you can calculate the population growth rate based on this population matrix. And that growth rate is called lambda. And if lambda is 1, then that means that the population is staying stable. If the growth rate is less than 1, then the population is declining. If the growth rate is above 1, then the population is growing. So when I put together this population matrix for all of the different remnants that I studied and calculated the population growth rate, it came out to be less than 1, 0.886. And the confidence interval did not overlap with 1, so this suggests that these populations are declining. Even though this is a relatively common prairie plant, these populations are in decline based on this model. So remember that I said that the number of seedlings that was produced in 2010 was only about 1/4 of a seedling per flower. And in other years, we have found much higher fecundity rates. In 2008, the rate of seedlings per flowering plant was about 3 times higher. So I wondered if I just changed out this fecundity rate, if that would change the overall population growth rate. So maybe it was just a bad year in terms of seedlings. So I replaced the 0.24 with 0.76, and and I calculated the growth rate with these fecundities, and I found that it didn't really make that much difference if I put in these higher seedling production rates. So apparently the number of seedlings produced doesn't have that large of an effect on the population growth rate. And in fact, if you make what's known as an elasticity table, this shows you the contribution of each of these transitions to the overall population growth rate. And you can see that the production of seedlings, that's a relatively low number, say compared to the number of flowering plants that stay as flowering plants, and compared to these small basal plants that stay as basal plants. So these are the 2 largest elasticities, or the largest contributions to that growth rate. Maybe I better say this more clearly. An elasticity means that if I change this transition here, a high elasticity means that that particular transition has a large effect on the population growth rate. So the 2 largest elasticities, again, are whether the proportion of flowering plants that remain as flowering plants the following year, or come back as flowering plants again the following year, and the proportion of small basal plants that survive and come back as basal plants. And relatively small contributions are the proportion of seedlings that are produced by each flowering plant, and surprisingly, the survival of those seedlings from seedling to basal plant. So we know that that has to be important, but it has rather a smaller effect. And all of these other survival rates seem to have a stronger effect on the population growth rate. My other question related to this is, does the remnant size matter? So are the smaller populations declining, and maybe the larger populations, maybe they're doing better? So what I did is I took my overall data for all the sites and I divided into two subsets, with the largest populations in one subset and the smallest populations, or the smaller populations, in the other subsets. And I made a separate population matrix for those two subsets. And I found that the larger population matrix did have a higher population growth rate, a higher lambda, and the confidence interval almost overlapped with 1. Still didn't overlap with 1, so it still looks like even the large populations are declining. But the small populations are declining much faster, and you can see that these confidence intervals do not overlap. And so there is a significant difference between the large population growth rate and the small population growth rate. And based on some randomization testing that I did, the probability of getting this much of a difference between between two matrices, if I just randomly sampled from all of my data, that probability was very low. So it looks like the small populations are declining faster. So what transitions, what part of the life cycle is important to those differences in lambda? So this table shows the contributions to the difference in lambda. And so a positive number would show that the large populations had a higher rate, say, of flowering compared to the smaller populations. A negative number, like what we see down here, would mean that the large populations had actually a lower rate of these flowering plants coming back as flowering plants. So you can see we have a couple of fairly large negatives, but most of the others are positive or kind of negligible. And so that these large positives, like the high survival of these flowering plants and the higher seedling production of these flowering plants, and then plus all of these higher survival rates for these various different basal plants, those overcompensated for these negative numbers that we see. And just to kind of show this graphically, we've got these low numbers for flowering plants that stayed as flowering plants, but all of these positive numbers that kind of make up for these negative numbers. So higher survival of all these basal plants and a higher production of seedlings, and notice over here, a higher survival rate of seedlings contributes to a higher population growth rate in the larger populations compared to the smaller populations. So looking at it this way, it looks like the seedling stage is important. It is important how much reproduction that they're having. And also these survival rates are also really important, so that these plants can survive long enough so that they can reproduce. So conclusions from this first question about population growth rate is that these fragmented Echinacea populations are indeed predicted to decline if things don't change, and that the remnant size matters, that the smaller populations are declining faster than the larger populations. And despite low elasticities, the seedling stage does make a difference if we compare the large and small populations and the rate at which they are declining. Moving on now to the second aspect of my research, and this is whether genetic declines that we see in small populations can be remedied by outcrossing between populations. And so I hypothesized that if these populations are genetically less diverse and suffer from inbreeding depression, that if I cross plants within a remnant, that those resulting offspring might not do as well as if I did, as if the population was larger and less genetically deprived. And secondly, I hypothesized that for these small populations, if I did do a between population cross, that those offspring would do better than the within population crosses within a small population. And then secondarily, I wanted to know, primarily I looked at kind of maternal plants, but secondarily I looked at whether the population size of the pollen donor mattered. So crossing experiments often show that if you have a cross between two different populations, two genetically distinct populations, that oftentimes fitness is improved in those offspring. And we call that heterosis, an improved fitness in the offspring of a cross. However, some studies have also shown that if you have a cross between populations that are too dissimilar, then that can cause outbreeding depression, which is when the results of a cross are less fit than the results of a, say, a within population cross. So we didn't really know what was going to happen with Echinacea, and so we decided to, actually we had done some previous experiments with Echinacea, but so this was kind of a follow-up experiment. We do know that there was inbreeding depression in some previous crosses that were done with Echinacea, but in those crosses, the seedlings were grown in a greenhouse and then planted into the field as plugs. And so we kind of bypassed the seedling stage. And so I wanted to look at, I wanted to make sure we included the seedling stage. So I got a little bit ahead of myself. We also know that there is some genetic differentiation between the Echinacea remnants that we study based on some work by Stewart and Eric Lonsdorf. And we have seen that there's a decreased genetic variation in the small compared to the large populations. And so this seems like it's correlated with some inbreeding depression in within population crosses. So again, I used plants from these various remnants, and Stewart had collected some, this is Stewart Regenius, and he collected some Echinacea seeds from some of these remnants in 1995 and 1996 and planted them in an experimental plot in 1996 and 1997. So now, by the time I was doing my crossing experiment in 2008, these plants had grown to maturity and were flowering. And a great aspect of this experiment that I was able to do is that these plants had all grown in a common environment, what we call a common garden. And I will also point out on this map, here is Hegg Lake. And so what I did is I did the crosses within this experimental plot, and then I collected the seeds from the crosses, and then I planted them down here in a experimental plot in this Hegg Lake wildlife management area. So these were the parental populations that were the source for my experiment, and I have them organized here basically from smaller to larger. And so the Stephenson Prairie is by far the largest population in our study area, and Northwest Landfill is kind of on the smaller side. And for each maternal plant, I would cross 1/2 of the florets on a flowering head with pollen that came from another population. Typically, I used pollen from the largest Stephenson Prairie population. And then the other 1/2 of the florets on that flowering head would be populated or would be pollinated with pollen from that same population. And then if there was a second head on the plant, then I would use pollen from a smaller population to see if the population size of the pollen donor made a difference. So this was the realized design, the number of maternal plants that I actually ended up having for all of these different populations. And once I had made the crosses and once the seeds were ripe, I took these achenes to Chicago, to the Chicago Botanic Garden, and I weighed all of these achenes. And you can see that there's a lot of variation in the weight of the achenes, and most of them are pretty light. And so the ones that are less than about 0.002 grams likely did not have embryos, and that's why they were so light. So these ones were the ones that probably had viable embryos. And so I just kind of made my cutoff for what I called seed set, which would be a successful pollination that resulted in a seed. I made that cutoff to be right at 2 milligrams. And so there were about 15,500 achenes that actually had a mass greater than 2 milligrams from all those various crosses. And so this is a graph that shows the proportion of total achenes that were actually successfully pollinated, the proportion of achenes that actually were seeds based on that weight measurement. And so you can see that my results varied by population. So the population was statistically significant as I analyzed this. The cross type was not significant. So some of the within population crosses, which are shown in black, some of these within population crosses had much higher seed set than the between population crosses, but in other populations, they had lower seed set than the between population crosses. And so there was a lot of variability. And so there was a difference. There was a significant maternal population by cross type interaction because we didn't always see the same trend in the different populations. So then I went ahead and sowed these seeds in the fall of 2008. And you can see over here, there's a nail. So I marked the sowing locations with nails and tried to sow the seeds kind of in a straight line to the west of the nails. And then we went back in 2009 and we were able to find quite a number of seedlings. And we marked them with little party toothpicks, which you can see here, so that we could find them from year to year and track their survival. So then in 2011, I did an Aster analysis of the overall fitness of these seedlings over their life cycle up to 2011. And I have continued to follow these plants, but I haven't updated my analysis since 2011. And at this stage, again, there is a significant effect of maternal population. Again, the cross type is non-significant because we didn't see the same trend in all of the different populations in terms of the difference between within population and between population crosses. But there is a significant maternal population by cross type interaction. And so this is characterized by some of these smaller populations having a lower fitness for the within population crosses compared to the between population crosses. And this might be expected if we're seeing genetic rescue here. And if these small populations are genetically depleted, we might expect lower fitness with the within population cross and a higher fitness with the between population cross. However, some of the populations, and especially this largest population, the between population cross actually had a lower fitness than the within population cross, suggesting outbreeding depression. So this would be the Aster prediction if I started with 25 Echines, which is the average number of Echines at each sowing position within my plot. And then I decided to look at the effect if I included the secondary crosses. And again, we have the same significant maternal population effect. Some maternal populations had much higher fitness and others had much lower fitness overall. And then again, this maternal population by cross type interaction. And you can see that there's no consistent effect of this between population cross using a smaller population. We can still see that the strongest indication of heterosis that we have is for these two smaller populations, especially for Northwest landfill. And then several of these populations look like they actually do better with the within population crosses. So some conclusions. Differential responses for within population versus between population crosses, depending on the populations crossed, depending on the life cycle stage. And it seems that small Echinacea populations may be candidates for genetic rescue by doing some outcrossing with bringing in pollen from other populations. However, there is some risk of outbreeding, outcrossing depression or outbreeding depression. So I would recommend to be cautious about trying this outcrossing, but it might be an option for very small populations. Now, moving on to question 3. Are Echinacea populations locally adapted? And so what I was looking for here is whether if I did a transplant, or if you were to collect seed from a long distance away and plant it into your prairie restoration, would these plants do as well as local plants? So a common guideline for prairie restorations is that we use local seed. And the reasoning behind this is that local seeds are thought to be well adapted to local conditions. And also we don't want to bring in seeds from a long ways away and then have them grow in our local area and then breed with natives and cause outbreeding depression. So we want to avoid that. However, small local populations may not have sufficient genetic diversity to survive long term. And so we also know that local adaptation varies amongst species. And so I just wanted to know part of kind of what's the answer for Echinacea, or to try to uncover some of that for Echinacea. So here's a map that shows the states in which Echinacea angustifolia is found. It's all the way from Canada to Texas. And then the smaller, or the lighter green squares or shapes are the counties where it is found. And so what I did is I collected some seeds from near our study area in west central Minnesota. And then I also collected some seeds from north central South Dakota and from northwestern South Dakota. And I planted all of these different kinds of seeds back into all three locations. So I did a reciprocal sowing experiment. I specifically tried to compare these sites because I specifically used an east-west transect because I wanted to minimize the day length differences that these plants would experience, and also I wanted them to have approximately the same kind of winter conditions. But still, there is a big precipitation gradient that could affect these plants. So plants growing out west would have much less annual precipitation, about 16 inches or 41 centimeters compared to 24 inches or 62 centimeters in Minnesota. So I collected seeds from 50 different plants at each site, and then I planted seeds from each site back into each of my sowing sites. And I had about 22,000 echines that I planted in 3 experimental sites. And so what I was looking for is a source by sowing site interaction where western South Dakota plants would be expected to have a higher number of seedlings if I planted these seeds in western South Dakota, and lower in Minnesota. And similarly, the Minnesota plants, or the Minnesota seeds, I would expect to do best in Minnesota and less well farther west. And that's not the pattern that I got at all. So in terms of seedling emergence, it just seemed like the main thing that mattered is where I planted them. So there was the highest emergence for all 3 populations in Minnesota and the lowest emergence in western South Dakota, as might be expected based on the precipitation gradient. The source of the seeds also did matter because the central South Dakota seeds didn't do very well at any of the 3 sites. Not really sure why that was, but it might be that I collected those seeds a little bit immature, and they just might not have had as much provisioning in those seeds. But notice that the source by sow site interaction was not significant, and basically all 3 sow sites, the seeds, the pattern was kind of the same at all 3 sow sites. So, no evidence of local adaptation based on seedling emergence. However, over the first summer, there was a difference in survival in the 3 different populations, where there was a trend that the Minnesota seedlings did not survive as well as the western South Dakota. And this was marginally significant. The source was marginally significant. The sow site was significant. There was higher mortality in central South Dakota, and I attribute much of this to the fact that there was denser vegetation in this central South Dakota site Both the Minnesota and the Western South Dakota site had been burned the year before I planted, so there was much less competition. Also, sadly, this Central South Dakota site got sprayed with an herbicide towards the end of that first summer, and that killed all the Echinacea in that plot. So I actually went out and did this survival census after that spraying, and so I found a lot of wilty plants, and I thought that maybe I captured all of their survival, but it's possible that some of them I just didn't find because they'd been killed by the herbicide. So again, no evidence of local adaptation because all three sites had a similar trend of the three different seed source populations. But what about long-term? Would the survival long-term be affected by where they came from? And what about flowering? So I did an Aster analysis a year ago that included all of my data up through 2019. And these plants were basal for several years, and finally some of them started to flower. The first flowers were only in the Western South Dakota plot, and the first plants to flower were the Western South Dakota plants in the Western South Dakota plots. In 2018, I think, is when I first saw a flowering plant in Minnesota. And then in 2019, again, I did not have any flowering plants in Minnesota. Or I take that back. In 2019 is the first time I saw a flowering plant in Minnesota. So after the 5th year, I started seeing an indication of local adaptation. And so you can see that these Western South Dakota plants had a higher fitness, and this was based on the leaf number, the total leaf number after 7 years. And so you can see that the Western South Dakota plants had a higher fitness in Western South Dakota and a lower fitness in Minnesota. And in contrast, the Minnesota plants had a higher fitness in Minnesota and a lower fitness in Western South Dakota. And so you can see this kind of crossed line pattern that you would expect with this local adaptation effect. A couple years later, after they started flowering, I was able to do an analysis that included flowering. So this was one of the first 3 plants that flowered out in my Western South Dakota plot. And so based on the data through year 2019, again, we have a higher fitness overall of the Western South Dakota plants in Western South Dakota compared to Minnesota. There still are very few plants that have ever flowered in the Minnesota plant. And so so our source by sow-site interaction based on the total number of heads produced is only marginally significant, but we are still kind of seeing this trend that at least in the Western South Dakota plot, the Western plants seem to be doing better than plants from Central South Dakota and Western South Dakota. Now, this analysis is, I used as my starting point the sowing of the achenes, and so this analysis, there's a strong effect of whether those achenes came up in that first year. So I did another analysis where I started with the assumption that that there's like the same number of seedlings from each plant. And so in this one, there is a significant source by sow-site interaction. And this is characterized by kind of a higher survival of the Western South Dakota plants and a lower survival of the Minnesota plants in the western South Dakota plot, and in contrast, just a barely higher survival and flowering production in these Minnesota compared to the western South Dakota plants. So it's not very obvious, but it is a significant interaction. So this suggests that there is some local adaptation in these Echinacea populations. So it's the same species across that range, but it does seem like it does make a difference where these plants are coming from in their ability to survive and their ability to flower. So conclusions, early on, I saw no evidence of local adaptation in the seedling emergence, although it was important where I got those seeds. But I did start seeing local adaptation, certainly in the survival of the basal plants, and also in the flowering of those plants. So as I mentioned, the first plants and still the most plants that have flowered are the western South Dakota plants, and almost all of the flowering has been out in western South Dakota. Now, putting all of this together, all three aspects of my experiments, what are some overall implications that I think we can derive from this research? One is that we definitely see a decline in these small remnant populations, even though as you're driving along on the road, you can often see Echinacea, and so it's not obvious that Echinacea is in trouble. But my research has shown that these populations, even though they're abundant, they are predicted to decline, and that there are some negative effects of genetic declines. Second overall implication is that small populations may be candidates for genetic rescue, for some bringing in pollen from other populations, or maybe bringing seeds from one small population to another small population. When those plants grow up, maybe they will kind of rejuvenate that population. However, there is risk of outbreeding depression if we do too much of that, so I would be cautious about that. And then also there is evidence of local adaptation, at least at the long distance that I used. So again, I would be cautious about transporting seeds a long distance to use in restoration. So clearly I had a lot of help with this research, and so I will just highlight for this particular presentation that I had a ton of help from Team Echinacea, and so you can see 1 year's team shown here, and just lots of other help and support. So that's my research. Thank you very much for your attention.